Everyone, welcome to Get My Class. So let's solve this question here. It says a typical nucleosome contains dash of DNA. Option A 150 base pairs, option B 200 base pairs, option C 180 base pairs, and option D 220 base pairs. So what does the question say? It says a typical nucleosome contains dash of DNA. So what are they talking about? Since in all the options they have mentioned about base pairs, of course we are talking about how many base pairs are present in a typical nucleosome. Okay. Now, what do we mean by nucleosome? What do you mean by nucleosome? Nucleosome, we know it is one of the level of organization in chromosome, right? Level of organization. Okay. So when we say level of organization, you would have seen how the chromosome looks. It is actually tightly wound, and it is not only DNA; it is the combination of DNA and protein in majority, right? Now, when we actually open up the DNA, you come across something called as a nucleosome, right? And what is nucleosome? Nucleosome is a bead-like structure, and which is made up of DNA as well as histone proteins. Okay, so what you can see here, these are histone proteins. Okay, there are around 8 molecules of histone proteins which form the core protein. What is this core protein? Around this core of histone, the DNA is wound. Okay, okay, and this DNA, which is wound around, is about 200 base pairs. How many base pairs? 200 base pairs. And at the end, it is linked by a linker protein, which is called as the H1 histone. Okay. So, what does the question say? The question says a typical nucleosome contains dash of DNA. So, how many base pairs of the DNA? That is 200 base pairs. Now, one more important point. Like how is that DNA is able to bind to these proteins? Because histone proteins, these are basic in nature. Okay. And what about DNA? DNA is acidic in nature. And what is the full form of DNA? The full form of DNA is deoxyribonucleic acid. So it is acidic. And the histone proteins are basic in nature. And that is the compatibility between this DNA and histone proteins, that they are easily able to attach to each other. Okay. And since in the question, what are they asking? They're asking about this particular stretch of the DNA, like how many base pairs are present in this particular stretch of DNA, which is wound around the histone 4 protein until it ends here and it is bound with H1 histone protein. So till here, so how many base pairs are there? Totally 200, on an average 200 base pairs. So let us see. Option A says 150 base pairs. No, it becomes very less according to the size of the histone protein. Then option B says 200 base pairs, so that is correct. Option C says 180, again which is slightly less. Option D says 220, which is slightly more. So which is the correct answer? The correct answer is option B, that is 200 base pairs. I hope you understood the explanation. Thank you.